Welcome, everybody. We are so happy to see you here. And people are still joining, but I will just start. This is a gene switch webinar on feed and genomics. So we are trying to seek what's the perfect mix. And we are happy that you're here to listen to us and share your ideas, of course. My name is Chala. I'm working for the European Forum of Farm Animal Breeders. So we are representing the animal breeding sector. And this is the program of today. So first I will give the floor to Elisabetta so that she can talk about the gene switch project, which she's coordinating. And then we will give the floor to Mario on improving predictive models for genomic selection. And then we will pass on to Jerry on microbes, metabolites, and pig health. And then we will have a panel discussion led by Johan van Arendonk from Hendrix Genetics and Filippo Miglior from University of Guelph and Lactanet, Canada. There are some house rules, so please, while you're not speaking, mute your microphones. And if you have any questions to the speakers, because we have question times, and also during the panel discussion, either raise your hand from reactions, or please ask it in the chat box. Please note that the webinar will be recorded and shared on our website and social media. So there will be some photos taken. If you don't want to be included there, please turn off your cameras. If there are any technical issues during the meeting, please write to the chat box or send an email to John Zurmendi, my colleague. And yes, so let's begin. Please, Elisabetta, the floor is yours. Maybe while you're starting, I can just share our first poll. So we have the breeding sector, feed sector, and mostly from researchers, and we have students and other participants. I don't know if you saw it, but this is the results. So, Elisabetta, the floor is yours.
Okay, so this is to give you an introduction of gene switch, the regulatory genome of swine and chicken, functional annotation during development. I'm coordinating this project with the help of Herbia Klock and Camille Benard, also from INRAE and from INRAE Transfer. So now we'd like to know why it's not advancing. Well, the consortium is made of 11 partners that you see shown in the slides. They cover 8 European countries, and we are also connected with direct links for the operations that we do, the ongoing work, with additional animal breeding companies. Gene switch has an ambitious global aim, which is made of 2 components. We want to deliver underpinning knowledge on the pig and chicken genomes and to enable its translation to the pig and poultry sector. So it's really a step forward, the simple annotation of genomes. We started in July 2019, sorry. So work is ongoing, and as you may have heard in other presentations or events, we are also connected to other H2020 projects and expanding our aims in the frame of Eurofund. You see the website as a reference. So gene switch is made of 3 specific and interconnected aims, A, B, and C. The slide is pretty complex, but I invite you to go to see more details on our websites. I will go very briefly on each of them. So A is functional annotations across tissues and developmental stages for both species, spanning early organogenesis, late organogenesis, newborn animals, and adults through existing data mainly. B is for using the functional annotations for precision animal breeding. It's the focus of today. And C is about standardization of data and processes, dissemination and outreach, which is another main pillar of this project. About aim A, what are we doing? Essentially, we want to identify the functional elements that are activated, poised, repressed in the chicken and pig genomes across the 3 developmental stages that you have seen. And the idea is to characterize their dynamics, that we call switches, and tissue specificity, as well as their patterns of conservation and variation. Of course, it is implicit in the project is an interesting approach of comparative genomics of mammals versus birds. And we want to deliver high quality, richly annotated genome annotation maps for these species that will be available to the whole community. The underlying aim for moving from aim A to aim B is the question, what's the role of functional genomic elements in the determination of the phenotypes of the adult animal? And of course, here we will gain new knowledge, especially of those elements that are active, poised, repressed during development. And of course, the main question as well is what's the relative impact of the genetic variation of these elements on production traits. So we go to aim B. And here I just restrict my words to the main questions or contents, because this is the focus of this webinar and will be illustrated by my colleagues. So here for this aim of precision animal breeding, we had in mind two different approaches. One is of the integration of genetic data. The other one is of integration of epigenetic data, which is more explorative. On the left side, the integration of genetic data, the question is, can functional annotations enhance the prediction accuracy of breeding values in commercial populations? And for the right side, we want to provide the basis for future studies focused on better farm management, for example, using lower quality and more sustainable feed. And about AIM-C is a quite rich pillar of the project as well. It's about standardization of data and processes, dissemination and outreach. We work in a fair way for findable, accessible, interoperable, and reusable data. For coordination, curation, validation, archiving, and so on, together with the European Biological Archives and, of course, the FANG data portal, which is our main main data portal for the data. We do extensive dissemination, communication, and training activities to ensure mutual interactions with all stakeholders. This event is one of them. And as I mentioned before, we are part of Eurofang that was established together with other ongoing H2020 projects like Aquafang, Bobreg, Geronimo, and Rumigen to facilitate collaboration and foster more research. So what is impact that we expect from these components that I've illustrated? First, high quality reference annotation maps for the whole research community that will be, I think, a great resource for both academic and applied research. We will deliver cutting-edge research results that will pave the way for further studies and also, we think, strategies. And we think there will be a number of European stakeholders' benefits, an increased understanding of the value of functional genome annotation to face current and future challenges for improving, for achieving sustainable productions. And this is my last slides. I wanted just to remind all of you, for those who come to Rotterdam, to attend also to our talks that I put in here for each project aim. So there will be one for project aim A, which will be a big summary of what we do for the annotation and the analysis so far in pig and chicken. There will be 4 presentations for aim B that you see listed here, and you will hear more today as well. And I wanted also to remind that under aim C, on 3 July, on the 3rd of July, in Rotterdam, there will be a nice event, I think, that is a joint event of Gene Switch with Geronimo that is also focused on chicken and pigs, Bitcoin or genomics, which brings more. And before leaving you, I'm told that I should alert you that there will be 2 poll questions shown right now on the screen. And otherwise, I close here and I give the floor to my colleague. Thank you.
So, Mario, maybe you can already interpret this somehow. So because 54% is not yet convinced on functional genome annotations that will increase genomic prediction accuracy. And maybe I can.
Either way, the pressure is on.
Yes. So I will launch the second question. I hope you will see it. Functional genome annotations will be most beneficial for complex traits like feed efficiency. So at least the majority thinks yes, but still 37% not yet convinced. So yes, the floor is yours, Mario.
Okay, thank you. Welcome all to my presentation today about work package 4 of GeneSwitch. My name is Mario Calus and I work at Wageningen University as an associate professor in the animal breeding and genomics group. My talk is about work package 4 and is entitled Improving Predictive Models for Genomic Selection. So just a quick outline to my talk. I'll briefly start with explaining what genomic prediction is for those of you unfamiliar with it. Then I'll introduce our work package, share some results and output so far. And then I'll end with listing some anticipated impacts and some take-home messages. So just very briefly, what is genomic prediction? Now put yourself in the position of an animal breeder, in this case a pig breeder who has a litter of pigs. And let's assume there's 4 litter mates in this case. And the question is, of these young animals, which of those are should be kept for breeding purposes? So in other words, which of those animals are better than the average of this family? So if they're really young, you don't have any performance information of these animals themselves. So you really can't tell those full SIPs apart. However, with genomic prediction, what you do is you take a DNA sample of each one of them. This can be through tissue, or it could be taken as blood. With this sample, you build a DNA profile of this individual, which consists of so-called SNP genotypes. And based on this profile, you can actually predict a genomic breeding value. So in this case, let's assume we focus on the trait number of piglets. And then what we find that this genomic breeding value tells that 2 of them have a positive breeding value and 2 have a negative one. So then to finish off the job, what you do, you select the ones with the highest breeding values and you keep those for breeding purposes and you don't keep the ones that have a breeding value which aren't good enough. So that's really in a nutshell how genomic prediction works, and it hopefully it clarifies how this technique accelerates animal breeding, simply because you can select the animals at a very young age before they've actually shown any performance whatsoever themselves. In work package 4 of GeneSwitch, the main aim is to extend traditional or state-of-the-art genomic prediction models to exploit the new annotation maps that for pigs and chicken that are generated within the project. And we try to achieve that first of all by developing models that can actually incorporate this additional information. But we also within the work package we generate FineMap QTL and eQTL ourselves that will also further enrich the information in the model. And then finally, we will validate those models in large-scale commercial data. So if we put this in an overview to show what's happening in work package 4, so everything inside the green boxes is happening in work package 4. So the traditional genomic prediction models, the input that they take are genotypes and phenotypes of a reference population. In this case, we're going to add 3 sources of information to this. It's first of all the functional annotation maps generated by work package 1 and 2. And then in addition, within the work package, QTL are being fine mapped for body weight in chickens. And also eQTL have been generated for pigs. So now, once we have all this information, this additional information in place, and also these newly developed or extended models, then we're going to combine this to validate the models. We're going to do that at 2 levels. The first one we call the level 1 validation, where we have 300 pigs available with whole genome sequence data, but also gene expression that's generated within the project. And the level 2 validation, that involves using commercial data from pigs and poultry. And I'll go into a bit more detail later, but there we have the traditional input to the genomic prediction model, and we're going to enrich that with the functional annotation to see if we can get higher prediction accuracies. So the work that we started out with is to work on different kinds of models. And one of the groups in INRAE has been working in particular on Bayesian models for genomic prediction. So to give a little bit of context, having this functional annotation sort of holds the promise that we should actually be able to better understand how complex traits work, because we can pinpoint certain regions and the genome better with it. So in this sense, it can actually help to do QTL mapping and to make inferences about the background of the traits. Now, once that's the case, we also hope that it can improve prediction so that it can give higher genomic prediction accuracy. So the starting point here was to use state-of-the-art Bayesian models. One is called Bayes-R and one is called Bayes-RC. I'll go in a bit more detail in a minute. And then to add functional annotation and multiple sources thereof as a prior information in the model. So to try and guide you through this step by step, here at the very bottom, we see a genotype of an individual, which consists of 0, 1s, and 2s. These are just allele counts of a certain allele at different positions in the genome. If we have those genotypes for a large number of individuals, and we have phenotypes, so measured performance, then we can use that to predict genomic breeding values for animals for which we have genotypes only. Now, so then one of the starting points is this Bayes-R model. And what Bayes-R actually does is it looks through each of those SNPs and it divides them in 4 different categories. There's a null category, which is assumed to explain no genetic variants at all. And then there's 3 that explain increasingly more genetic variants. So there's a small fraction of the loci is being allocated to this high category. Now, a previous advancement of the Bayes-R model was to actually do this division in the 4 groups within different groups of annotations. So here on top of the genotypes, 2 levels of annotations have been added. The first one were QTLs that have been recorded in the animal QTL database. And the other one are GWAS hits. And then the idea is that if there's a GWAS hit at a certain locus, then that locus is put in the first category. Now, if there's a QTL hit in the animal QTL database, then that locus is put in the second category, and all remaining loci are together put in a third category. And with any...
So I think Mario explained about these 3 different categories. And then I think that the next step, what the group at INRA did, is the instead of assigning a SNP to a single class, they also extended the model to have a multiple annotations possible for their SNPs. And then again, to allow for different ages of the development stage of the animals, but also the different tissues. And this is all, I think the main message is that this software has been developed, but also been made publicly available on this GitHub address. So, okay. And I think they also applied it to a study with not data from gene switch, but from other data from INRA, from other projects, where they showed that also their annotations can have an impact on the accuracy. I can't give you all the details because I was not too involved in preparing this slide. Apologies for that. And also for daily weight gain, that they use these kind of annotations. And Mario is back.
Here, as you can see, is for the first trait, there's actually an advantage of using these annotations, a small advantage for average daily weight gain, but there isn't for back fat thickness of using annotation in these new models. And the annotation here was based on gene function. So it was based on QTLs and the category or the trait it was associated with. And you can see here that, well, for average daily weight gain, those that are associated with the feed conversion, they have the highest proportion of loci that are assigned to the class that explains most variants, which sort of intuitively makes sense. On the other hand, the differences between the trait categories is not that big. So in the previous slide, we used data that was already available at INRA of pigs. And then, well, one of the things in the project is that the data that's coming available in the project is not there until the third or the end of the third year of the project. So to start working with the models we developed, we also started to work with available data on the diversity outbred mouse. And just to say a little bit how this population looks like, so we had here something like 4 generations of data of outbred mice that derived back 10 generations ago to 8 inbred founder strains. The interesting thing about this data is that very many different traits have been recorded on it. So what we did is we applied GBLUP, which is the straightforward linear genomic prediction model, to a number of traits, but we also applied a machine learning model because our aim here was actually to develop machine learning models for genomic prediction. And these first 7 traits here, they're all polygenic in the sense that they are affected by very many loci and each one with a very small effect. And what you can see is that the prediction accuracy along the y-axis is typically somewhat higher for the standard linear model, or at least as good as for the machine learning model. So in some cases, the linear model is actually quite a bit higher. But interestingly, in addition to those polygenic traits, there was also a number of traits that were known to be affected by epistatic interactions. And when we looked at those, what we found is actually that the machine learning model had a higher prediction accuracy than the linear model, which fitted the expectations because machine learning models are more flexible and they should implicitly be able to model interactions between loci. So this really seems to be the case that this works. So that was a really nice result. Then in the next step in the analysis on this mouse data, we also added gene expression data of these individuals. And then what we did again analysis for a large number of traits, but I'm just showing you here our results for body weight, which was measured at 10, 15, or 20 weeks of age. And first we ran a standard GBLUP model just using SNP information. And you can see that at all of these time points, the amount of explained variance by the SNPs is about 40%. Then we fitted only gene expression in the model. And it's good to note that the gene expression was measured at 26 weeks of age. So it was measured after the last body weight trait. And 2 things to note here. First of all, the gene expression explains more phenotypic variants than the SNP genotypes do. This holds at all the time points. And phenotypic variance explained is highest at this moment in time that is closest to where the gene expression was measured. So here, body weight at 20 weeks was closest to the gene expression being measured at 26 weeks of age. And you can see that most variance was explained here. So then finally, we constructed a model that took both SNPs and gene expression in at the same time. And we were able to tune the model such that the variance explained by the SNPs was rather stable on the different time points, but still the variance explained by the gene expression was increasing if the time between measuring the gene expression and the phenotype was shorter. So that's about the model development so far. Then a little bit about the fine mapping activity in chickens for body weight. So here the group of Urian Kailborg at Uppsala University is using data from the Virginia chicken lines, and these lines, they're very interesting in the sense that for 60 years they've been doing bidirectional selection for body weight. So there's been a line where continuously body weight was increased by selection, and the other one it was continuously decreased. And at some moment in time, the two divergent lines were crossed with each other to make an advanced intercross line. And this gives a unique design, but also a very good resource with high resolution to fine map QTL. And to date, more than 20 replicated loci have been found that together explain more than 50% of the genetic variants. So that's about the fine mapping of the QTL. And then another activity is on measuring gene expression and then finding eQTLs in pigs. For this activity, in 3 different places, in each of those places, 100 pigs were sequenced, but also gene expression was measured on these individuals from 3 different tissues. So it was in the liver, the duodenum, and a particular muscle. So what's been done with this data so far is, first of all, the eQTL mapping, and there in total of 18 associations have been found that are consistent across the 3 different tissues. So there were very many eQTL found, but there's a total of 18 that are very consistent in all these different tissues. And what's currently is work in progress, but that is using this data for this level 1 validation, where we're actually going to do genomic prediction with this set of individuals. We have identified 10 different traits that are measured on all of them that we will use as phenotypes, but additionally, we will also use the gene expression levels as phenotypes, which gives us thousands of traits for these animals to work with. So then going from the level 1 validation to the level 2 validation. So as I said, the ultimate goal of the work package is to validate in commercial populations that what we have developed works and hopefully gives added prediction accuracy. So what we're going to use there is the gene switch data. So we have the new annotation maps. We'll have FIMAP QTL for chicken and eQTL for pigs. But the thing is that both of these, they are at base pair resolution. And the commercial data where we're going to do the validation with is very powerful in the sense that we'll have tens of thousands of individuals with genotypes and phenotypes, yet the genotypes are at 50,000 SNPs across the genome. So we have to make sure that the two can be matched together. So basically what we have to do is go from the 50K SNPs, genotypes that we have in commercial data, to a whole genome sequence, which basically means having millions of SNPs. And this will be done, this is currently ongoing, by imputing those genotypes up, such that in the end, we have for those tens of thousands of individuals, we have genotype data at the base pair resolution. And then we can proceed to do our validations. So what output have we generated so far? There's 2 published papers in G3. One is on the Bayesian models, one is on the machine learning models. And there's also 2 preprints, again, one on the Bayesian models and one on the machine learning models. And the latter includes also the results on the gene expression data. And the Bayesian models, they have been implemented in a package called BaseRCO, and that package is also freely available through GitHub. So to wrap up, what is the anticipated impact of Work Package 4? If we look at the results so far, the individual gene expression data seems very promising in the sense that more variance is explained with those compared to using SNP genotypes. And we also get a higher prediction accuracy with them if we include those in the model. On the other hand, we think that current costs, but also the sampling complexity, so when are you going to measure, take the samples and from which tissues to standardize all that, may prohibit implementation at this moment in time, but perhaps somewhere in the near future, costs at least will be quite a bit lower, and then perhaps this could be considered. And finally, the added value of using the annotation in the genomic prediction. So was nice to see in the poll up front that close to half of you think that it will give at least 10% more prediction accuracy, and the other ones say, let's wait and see. Now, we will hope to demonstrate it to you in the near future. But one thing that's important is that this annotation information, to make use of it, we have to go to whole genome sequence base pair resolution by imputation of the commercial data. And the other thing that we've been thinking is we have all these different annotation maps, and now we actually have those Bayesian models that can properly combine them. But for implementation, perhaps it would be better if we could actually integrate all these annotation maps, so that we get only like one single value for each locus in the genome. This would make implementation probably a lot easier. So the take-home messages: GeneSwitch generates new annotation maps and genomic prediction models to make best use of this new annotation information. So far, we've been focusing mostly on model development, and as a result, this BaseRCO package is now freely available on GitHub. Our results show that individual level gene expression explains a considerable variance, yet this depends on the moment of sampling. So the closer you get your gene expression to the moment where you want to predict your phenotype, the better it is. And current work in progress is the validation, the ongoing validation to show to what extent we can capitalize on the annotation maps requiring whole genome sequence resolution. So I would say the best is yet to come. So please stay tuned for our results in the next year and a half.
Yeah, thank you, Mario. Well, we're a little bit over time due to connection problems, but Barbara asked a question. But if there are any other questions, maybe we can take at least one, and maybe if anybody raises their hand. In the meantime, Marco replied to Barbara's question. If not, then maybe just to be on time, we can go to Jerry's presentation. I don't see any hands so far. Okay, thank you. Jerry, the floor is yours. If there are any other questions, you can still write in the chat box, and Mario, I think, can reply from the chat box together with Marco.
Okay, good afternoon, everybody. My name is Jerry Wells, and I'm going to be reporting on the progress in Work Package 5, which is about the influence of maternal diet on epigenetic programming in utero and offspring. And I've called this Microbes, Metabolites and Pig Health, because this is the key words which essentially capture the essence of what the talk is about. So in this work package, I should put this on presentation mode. Sorry. In this work package, yeah, we have performed a big gene bioenvironment study, and we are. Sorry, you can hear me, okay? Yeah.
Yes, no problem.
We generated the RNA-seq and ATAC-seq data from this study and all the biorepository of samples, and we are busy with analysis of the data and the integration of the data, which will be necessary for the full interpretation. Okay, so diet, microbiota and health. I'm sure everybody knows that changes in the microbiota have been associated with many human diseases, even those that don't seem to be directly involved in the gut. So there's an interplay between the host and the microbiota, which affects disease susceptibility, disease severity, and also treatment resistance. So antibiotic use, for example, or infections in animals is something that can also cause this dysbiosis, and also diet. So diet, and particularly the lack of fermentable fiber, which reaches the colon, is one of the reasons why it's hypothesized that humans have had an increase, population has said in the Western societies in particular, have had increasing occurrence of allergies, autoimmune disease, and also other diseases as well. So in the last decade, there's been a lot of research looking at diet microbiota research, and it's become very apparent that the metabolites produced by the microbiota, which is obviously connected with the diet, play a big role in human health. For example, some peptidoglycan fragments which cross the barrier are very important in enhancing systemic immunity and priming of macrophages and neutrophils, for example. The bottom one is a study about the aryl hydrocarbon receptor, which is a xenobiotic receptor. And metabolism of tryptophan generates metabolites that are recognized by this receptor. It's all kinds of functions in the host, depending on which cell types express the aryl hydrocarbon receptor, including gut barrier defense, including immunity, including immune regulation. And this is a study that we were involved in recently, showing the potential to use increased tryptophan in the diet in celiac patients to alleviate intestinal inflammation. At the start of the project, we wrote a review about the metabolites that we focus on in this project. At least we designed diets to deliver higher concentrations of these metabolites in the colon, and then in the circulation. And we are focusing on the short chain fatty acids. So just for definition, these are acetate, propionate, and butyrate. These are found in concentrations of up to 100 millimolar or more in the colon, in the feces. And usually acetate is about twice, present at twice the concentration of propionate and butyrate. And I'll tell you more about levels in the circulation later. But one of the things that has become very clear, and a lot of these studies are in mice. There are some studies also in humans, in diabetic patients with short chain fatty acid supplements, and those people in the seminar that come from the nutrition area will know also there is a big interest in using this in animals, in agriculture. So short chain fatty acids per se in the diet. And in humans, they have been shown, in mice, sorry, they've been shown to be important in epigenetic imprinting in bone marrow cells. So if you take germ-free mice and then you compare histone modifications in those with germ-free mice which have been recolonized with a normal fecal microbiota, or have been just administered those 3 short chain fatty acids, you see a lot of you see it almost recapitulates all of the epigenome effects that you see with a conventional microbiota. I won't say much more about these because we for time reasons we need to. But why short chain fatty acids? Just a few more papers from not in pigs but from the whole field. So in humans, in mouse models, sorry, acetate yielding diet imprints an antimicrobial program. So this could be important in infection. Acetate delivered in the drinking water has been shown to attenuate asthma in a mouse sensitization model, and it was also effective in the maternal diet. So that was just given in the maternal diet. That interested us a lot when we were writing this proposal, but also the short chain fatty acids have been shown to prevent the onset of type 1 diabetes. So this is not the obesity associated form of type 1 diabetes with metabolic syndrome, but the type 1 diabetes that is associated with some genetic factors as well as environment or lifestyle factors. So short chain fatty acids then. So how do mammals get exposed to short chain fatty acids, and what's the impact of the diet? Well, one thing that's happened in humans is that our fiber content in the diet, but not digestible fiber, but non-digestible fiber, has decreased dramatically compared to our ancestors several thousand years ago. And now it's considered relatively low compared to what it was then. And non-digestible fibers pass from the small intestine into the colon, and they're the more complex and higher numbers of microbes found in the colon are involved in fermentation of those polysaccharides. So they have many more enzymes to break down these complex carbohydrates. And as a byproduct of their metabolism, they produce short chain fatty acids. And these are what you find in the colon at concentrations, as I said, of say in humans, around 60 millimolar per gram of content of acetate, about half that as propionate, and a bit less as butyrate. So in the intestine, short chain fatty acids have been shown to have numerous roles in maintaining the gut barrier and interacting with the mucosal immune system there. So they can increase regulatory T cells, which helps control inflammation. They induce digestive hormone function through receptors on enteroendocrine cells. So these are a population of cells in the gut epithelium that produce hormones in response to metabolites. Short chain fatty acids are one of those metabolites that can induce hormones such as PYY for appetite, or these others. Some of these are also involved in like GLP-2 in increasing the surface area for absorption. Butyrate importantly also serves as a fuel for enterocytes. And it's one of the reasons probably that it's not transported at such high concentrations into the lamina propria. And they have direct effects on epithelial cells, inducing many gut defense molecules. So what's going on in the animal field? There are already indications that short chain fatty acids have quite profound effects in animals as well, and in pigs, which is the subject of Work Package 5 study. For example, the infusion of short chain fatty acids in the ileum increased carcass traits like meat quality, lipid metabolism of growing pigs. And in a review, it was concluded that short chain fatty acids could prevent fat deposition via regulating hormones and genes in these animals. So there is a bit more information also in pigs, but currently it's relatively sparse at the functional level. So these are sort of concentrations that you can find in serum and different parts of the intestinal content in pigs. And this comes from this paper I showed from Xiao et al. In Animal Nutrition. We've also measured them in feces on the pigs in different diets in Work Package 5, and we find similar levels. I'll tell you more about the differences and the effects of the diet in a moment. But in the colon, for example, acetic acid, 45 millimolar. This would be per liter of content, but if you in grams, it might be very similar to humans, about 60 millimolar per gram of content. Less propionic acid, and then butyric acid, about usually around 10 millimole per gram in our case, and also in humans. So quite similar to humans. There's much less short chain fatty acids in the ileum, and notably some of these, like butyric acid, is very low, and propionic acid. So it's mostly acetate produced by those facultative aerobes that are involved in the digestion or in the fermentation and competition with the host for carbohydrates there. Okay, so a little bit of introduction now to the effects that we are expecting to see of diets that will influence short chain fatty acid availability in pigs and epigenetics. So I'm sure people in this seminar are familiar with this, but DNA is wound up, compacted by histones, and these histones form a complex, and the acetylation of the protein tails or other kinds of modifications on these histones affect the compaction of the DNA, and that typically DNA which is more open and not compacted in these nucleosomes is more accessible and more active for gene expression. What this is just a summary of what I've said, that acetylation and methylation play important roles. Acetylation of the leads to more open chromatin and expression, whereas the deacetylation leads to more compaction. Butyrate and propionate are known to be inhibitors of class 1 histone deacetylase activity. I personally don't think they directly inhibit the histone deacetylases. They don't look like pharmacological inhibitors of these enzymes. I think it's more indirect, and perhaps through the AMP kinase or the other signaling mechanisms. Acetate is very likely to activate histone acetylases through effects on acetyl-CoA pool, which increases AMP kinase activity. And that's known, for example, to acetylate a number of histone deacetylases, but also histone acetylases. So that brings us to the aim of Work Package 5. So in this work package, we want to investigate the possible role of short-chain fatty acids as epigenetic regulators. And we focus on two different types of tissue, liver and muscle metabolism. The muscle makes a very strong link to the work I showed earlier, indicating that that infusion of acetate into the ileum, which is very different to what we do, had an effect on muscle mass and triglycerides in the blood and so on. So based on all this evidence and research that I've been telling you about, our hypotheses are that high fiber diets, which increase the concentration of acetate in particular, because butyrate is not taken up in very high amounts across the epithelium and going into the circulation, where it is the case for acetate, to some extent propionate as well. That these diets, through changes in the microbiota and fermentation, will increase the serum concentrations and the availability in the circulation to the piglets. We also wanted to explore whether these diets in the sows would have effects on the fetus in utero. And I'll show the plan for the experiment in a moment. Because evidence in mice is that short-chain fatty acids in the circulation can exchange in the placenta and then have effects on disease outcomes. We hypothesize they would also have epigenetic effects, but that has to be still shown. We also designed the experiment with the idea that if there was effects of these diets in the sows, that there might be some imprinting that will have phenotypic effects in the offspring for at least 8 weeks postpartum. And this is because of studies in rodent models. Serum acetate, we predicted, might regulate PPAR alpha and lipid metabolism, reducing lipogenesis and lipid triglycerides in the blood, so leaner, but also effects on muscle directly, increasing muscle mass through upregulating type 1 myosin, and increasing the oxidative capacity of muscle, meaning that it could use fats and other molecules, including short-chain fatty acids, for metabolism. And the muscle would also be able to take up higher amounts of glucose, so it would be more insulin sensitive. And it would affect also the endurance of muscle. So these are probably traits that are also very interesting for agri-production and animal health. This is an overview of work package 5. So initially we had a high fiber diet. So Francesc, who's in the seminar, is a nutritionist, and he helped design these diets based on what was known about short-chain fatty acids in the feces of pigs, fed different diets. We also had a high fiber diet, but included some sodium acetate in the diet. This would, short-chain fatty acids have been included in the diets. Acetate has also been included in human studies on insulin sensitivity in human subjects as a drink, and in mouse studies included in the water. And then we had group 3, which is, it says no fiber, but it's very low non-digestible fiber. So it would not be feeding those microbes in the colon that produce most of the short-chain fatty acids. And 2 months post-weaning, then in pregnancy 2 months post-weaning, we had 2 males, 2 females from each of the litters for making the biorepository. And in a second round or second parity of these cells, which remained on those diets, we had fetuses, 2 males, 2 females from day post-fertilization 70, also for the biorepository. The biorepository is a very interesting collection of tissues that we could look at for other effects of these short-chain fatty acids, but we focused on liver and skeletal muscle. Muscle because of the reasons I said, but liver also because it is an organ that metabolizes the short-chain fatty acids when they go from the portal vein, from the gut, and really reach high concentrations in the portal vein. They're metabolized by the liver. And it will be interesting to see what effects these have on liver metabolism and liver function on some of these other pathways, like lipogenesis, for example. So we have generated from liver and skeletal muscle, both from the piglets and from the fetuses, RNA-seq and ATAC-seq, and are now looking at the data analysis and integrated analysis. So what I can tell you about the diets is that we've had difficulties with technically measuring short-chain fatty acids in serum. They are not insurmountable, but technically that's taking us a lot longer to do. But we have measured short-chain fatty acids in feces recently, and we saw that these diets generally have the expected effects. So we will get data also on individual cells and also from the piglets, which can be used for the data analysis and integration of that data. So we can stratify those, the piglets somewhat. And the high fiber plus acetate generated the most acetate in the feces, the high fiber second, and the no fiber a lowest amount of acetate. These differences are about 2 to 3-fold, and yeah, they are at levels which in humans and in mouse studies have had physiological effects. So we hope, yeah, in the future to see more from this. So the take-home messages are that yeah, short-chain fatty acids are major players in the interplay between diet, microbiota, and health. From a nutritional point of view, there may be apart from bulking, there is not so much attention, I think, given to non-digestible fiber, but it does produce short-chain fatty acids that provide energy for the enterocytes and important functions in the gut, but then they go beyond the gut and impact on other organs, particularly acetate, which is quite high levels in the serum. From some NMR analysis, we see that, so NMR is a very laborious technique. It's too laborious to use for all the samples we have from serum. But we see that the acetate reaches concentrations over 200 micromole per mil of serum, and propionate in the order of 20 micromolar, and butyrate at the very low micromolar, which is actually quite similar to what's been seen in humans who are on high fiber, let's say healthy diets, eating lots of vegetables and other sources of fiber. So we've seen that the short-chain fatty acid concentrations in the colon can be modulated by inclusion of non-digestible fibers that get fermented. And we hope to have some more news about the outcome of the data analysis in the remaining part of the project. And don't forget to feed your own intestinal symbionts too, because it's undoubtedly important for human health. Lastly, I like to invite, acknowledge all the people involved in this study. So Elisabetta is the coordinator of GeneSwitch, along with Hervé as a deputy coordinator, but Elisabetta is also helping me very much with this cooperation in Work Package 5 and the running of Work Package 5, along with Hervé and the team at INRAE, Sarah, Andrea, and Shmana, and at Wageningen, it's Linda Lonen, Jos Buchholz, our bioinformatician, who's working with the bioinformaticians at INRAE. And also I'd like to thank Francesc Molist and his colleagues at Schothorst Feed Research, who were involved in running the animal study for us, and the collection of tissues along with Linda from our group. So thanks very much for listening.
Thank you, Jerry, for the interesting presentation. We have already 2 questions in the chat box.
Okay.
So the short-chain fatty acids are microbial macronutrients, says Yap. Can microbial micronutrients be as important or even more important?
Yeah, I mean the vitamins, of course, that are produced by the microbiota that are really important for us. Vitamin K, for example. Yes. So that's the example I know of for micronutrients.
Mm-hmm. And Reza asks, what is your opinion about adding short-chain fatty acids on diet and then following its effects?
Ask me what the opinion is.
Yeah.
I think it's a very interesting idea to also include them in the diet. The advantage of including them in the diet is this, that in order for short-chain fatty acids to get transported, they have to be transported across the epithelium. Passive immunization is is very slow in the colon. So we've tested this, in organoids, and they use transporters. But butyrate mostly gets metabolized. Propionate is transported, and acetate is the most highly transported short-chain fatty acid. If you give it in the diet, in the stomach, they will be proteinated, and I think the transport will be higher. So I think drinking water or in the diet as a sodium salt will result in higher uptake of short-chain fatty acids into the circulation. I mean, in the lamina propria, it happens and they go into the circulation because also the blood vessels there have transporters for this, but they have been little studied. But the transporters in the epithelium itself in the gut have been fairly well studied, but not in pigs yet.
Great,
so I think it's a very interesting idea, and it may actually be an alternative to giving non-digestible fiber, but I think you might not get the same health effects in the gut, which are also very important for the overall animal health. And using fiber probably supports a healthy microbiome and other metabolites as well that are beneficial for health, like the aryl hydrocarbon receptor agonists. But maybe Francesc, if he's in the audience, could say what he feels about the use of fiber in diets and the circular economy and so on.
Thanks, Jerry. But no, I agree with you. In this case, we were using the sow. That is an animal that usually we can incorporate already quite a bit of fiber in these diets, and the sow will benefit from the fermentation. So that's why, yeah, this trial was interested also to use the sow as a model to see, okay, when we add, in this case was sugar, beet pulp and pea fiber, and also we add some acetate directly to the diet to see what levels we can reach when we go to really high levels, because fiber will be a more common feedstuff in swine diet, as Jerry mentioned, because we need to use more byproducts from the food industry. So in this regard, it's also interesting to see when you go to these high levels of fiber, what you get back at blood level, and how the sow can use this fiber. So totally agree with Jerry that fiber will become more and more important.
Yeah, and certainly in the human area, I mean, there are huge meta studies showing a correlation between the intake of fiber, the fiber that reaches the colon and is fermented, and all kinds of diseases, including cancer and so on. So there's actually very good metadata that healthy diets containing lots of fiber have many health benefits for humans. And it will, although the pigs are not so long living, I think some of those health benefits, well, in the agri-production industry, I mean, some of those benefits will be short-term as well, on gut barrier, gut health, resistance to infection, immunity as well, and development of immunity in young animals.
Great, thanks. That was a extended response, very well understood, I think. Maybe we can move to the panel. So I would like to invite Johan and Filippo and give them the floor. And we can maybe also, we had 2 questions just before the panel, so maybe I can launch them while Johan and Filippo are opening their cameras, or if they are, I can see them yet. Yeah. So the first question was about breeding companies. They should spend more resources on collecting gene expression data. So your ideas about this?
While we are waiting for the results to come in, it's an honor for me to chair this panel. And it was also a particular pleasure for me to see 2 of my former colleagues at Wageningen University present excellent work, which is the result of teamwork. Okay, there's a lot of people that are now convinced, eh?
Yes.
But this is probably very similar to the. I would be interested to see how many of the people that are working at breeding companies voted yes here. But that's because it's always easier to shop with someone else's wallet. But that's. And you had a second question. You said.
Yes, I will just ask it now. So, breeding companies should spend more resources on improving functional genome annotations. Yes, with slight majority.
The results for most questions are pretty similar, I would say.
Yeah. Okay, the floor is yours.
Yeah. Thank you very much. So I think the objective of today's seminar is to, one, be informed on the outcome of this particular world package of GeneSwitch, because the program as such is even larger than what we've shared here. And then in the discussion to focus on, okay, how could this impact the practices in animal breeding? And if I'm not mistaken, Filippo has prepared the first reaction to that. Is that correct, Filippo?
Thank you, Johan. Good to see you, by the way.
Yeah.
It's a pleasure to be here, and thanks for the invitation, everybody. So, first disclaimer: I'm a dairy person, not a swine and poultry. But my first reaction when I saw that answer. Exactly, said. I think the majority here are more researchers and not fully breeding company staff, and would be nice to see that answer to that question stratified by who do you belong to. You know, researchers are always positive. Let's do this, let's do that. But then the bottom line for a business company is to see what is the value added of. Am I gonna be able to recuperate money on my investment and so on? So that was my first comment to you, Johan. And so I get the question. I'm gonna ask you back. Now you're a part of a breeding company, and maybe you will answer later this question. Maybe you will not answer. But is there enough evidence now to invest, or is there a risk not to invest for a breeding company? Even there's still a competitive environment. So what if one breeding company invests in this type of technology and another breeding company doesn't? What is the risk of not doing that, in a sense?
Yeah, in that sense, I don't think there's a lot of difference between a breeding company and an academic institution. There's always risk. If you don't investigate in certain research, then you might miss out. If you do invest in certain research, you miss out on others because there's not unlimited resources. I would have phrased the first question a bit differently, because for me, it's not a question whether functional annotation is going to help, because it will. The black box approach has brought us a long way, but of course we can do better. But the big question is when. Yes. Because well, I would say, and I realize it sounds negative. The promise is there for long, but when will it be realized? And well, you could say you need to be patient, but I would like to hear from the research community in this talk. What are the limiting steps now? Because I'm really impressed by the research that has been done, but you can only do so much. So what are the factors that will really bring that functional annotation is really going to improve? Well, how we, not only genetics, but if I listen to Jerry's talks, it is not only about genetics. It will tell us also a lot about how to best feed. So the feed genetics interaction is particularly after Jerry's talk, pretty obvious. But what are the rate limiting steps? And please don't say money, because my next question, if you would say money, is money for what? So please, what are the breakthroughs that you maybe already have, but it's just a matter of scale of what is it? I don't know. Or the moderator not supposed to ask questions?
No, absolutely.
Is there anyone who wants to respond?
About I can try. Yeah. About the value of functional annotations, and the breakthrough that you expect is that at some point, and in the case of gene switch, it will be fully delivered at the end of the project, you will have functional annotations of reference, including a lot of features for each portion of a genome that will be usable, shareable with the community as well as the breeding companies, I mean, whatever stakeholder. And those data can be used in many ways. I mean, of course, as it was exposed in genomic selection, I mean, predictive biology, so prediction models. Maybe, as Mario said, ideally also giving each locus a single value, which will be the result of all the integration of annotation that you can have. And then the outcome, the final outcome of this, maybe it's early to say that it will be a breakthrough, that you will triple the improvement that you can make to animals. I would not say that, but especially for some cases, and I'm thinking of very complex traits that are little known, and that should be improved also in the definition of the phenotypes, which is the other big portion of a deal for making genotype to phenotype. It could make a real difference. I mean, that's what I expect. But it's not yet there, of course.
No, I accept it. Mario, you also want to respond?
Yes. I think in the end it will come down to accuracy and resolution of the data at the different levels. So as I explained, what we will need to do is impute the genotypes of the sequence data and then match that with the functional annotation, which is at base pair resolution. So I think it's going to be really important that this sequence data that we have imputed, that this has very high accuracy, and also that the annotation data has high accuracy. And perhaps the main advantage could come for traits where there's not so much information available. So if the traditional genotypes and phenotypes are limited in the first place, that simply leaves a lot of room for improvement, and that's where this additional information could be most useful.
Okay.
So maybe to draw, you said it's not a question of, it's more a question of when are we going to capitalize on this, right? So maybe to draw an analogy, like years ago we did research on resilience, and then we couldn't really find something that was important. But in the last couple of years, some of our colleagues have been quite successful simply because they finally had the resolution of the data needed to pick things up. There was so many repeated measurements in time, and then finally we could pick it up. Whereas before, having monthly measurements or even less frequent, the resolution of the data simply wasn't there to find it.
Yeah, but there's also I understand what you say, and I also do agree with it. But there's also a bit of a chicken and egg story, because it's going to be most beneficial for traits where we know less. But if we know less, we have less data.
Yes, but we will have the functional annotation, which is not measured on the individuals where you measure your phenotypes and your genotypes. Okay. So that's external information that's already there. That's the idea.
Okay. Very good. Let me open up for other questions related to and as said, yes, if there are questions to the moderator or to Filippo, then that's fine. So please, if there's a question, if you don't know who to ask it for, then just ask it generally. If you have a specific person in mind, then invite that person to react.
There's a question from Jan.
Yeah. Which reads, can functional nutrients and metabolites intervene with functional genomics? Jerry, are you able and willing to react to that?
Yeah, I can give partly a response to it, I think. I mean, there is in vitro data showing that the short chain fatty acids that I mentioned do have epigenetic effects. And there's also evidence that this can be beyond the gut and in the body, for in the example of the airway, for example, and the immune system, and in the bone marrow, even for hematopoiesis. So they can affect the immune system. The big question, I think, is something we're tackling in Work Package 5, which, where are the thresholds? So if you have a low fiber diet and a certain amount of acetate, but then you increase that twofold with a diet containing more fiber, what sort of impact will that be? And is that going to have a physiological effect? And what will be the impact of animal genetics on the responses to these nutritionally and physiologically active molecules? So these are sort of big questions, I think. But I have to say that there are certainly very good indications that circulating levels of short chain fatty acids have health benefits in humans. As I said, there were these large meta-analysis of clinical trials with different types of diets and populations. There's also been studies in pregnant mothers with children at risk for allergy, and looking at the levels of short chain fatty acids in those mothers and the incidence of allergic problems, or any kind of respiratory problem or development of atopic eczema and allergies and so on. And there was a very strong correlation for acetate, despite it being a population cohort study with all the genetic variation that you get. So mothers with higher than median levels of acetate had significantly fewer visits to the doctor with their children and less problems. So yeah, I think at the population level there are effects, but the big question is, how big are they, and how much will genetics of the animals impact on that? And understanding if there is a relationship and how it can be exploited would be really interesting, but we're probably far away from that.
Yeah. Okay. Thank you.
We had a couple of questions. If there is nobody or no comment, maybe just to.
Yeah, I was going to use the questions that you did right in the briefing, if that's okay. So that's what could the consortium do? That would be my first, to increase the impact of its activities. Then I want to make a consortium be a complement for setting up a workshop like this. But I think I would encourage you to continue doing so. It's a very good initiative to be present at the World Congress, to start at this point in time already the interaction. So where's the pains of the industry, and where maybe some of the activities that you do, where they could help. And I'm sure that that's part of your milestones. That for sure would be one of my suggestions. And then the second question is what can sector actors, how can we make better use of your work, was another. Well, I guess also that one. Communication in an early stage, and please do not only rely on scientific publication, but interactions like this I think would help. Then I was a bit intrigued by the questions that were asked. Is what could breeding industry do to accelerate your research? And then on the one hand, it was about investing, but I would like to know what kind of data would you be in particular looking for, because we can start collecting gene expression data, but there's a big risk of garbage in, garbage out, that we collected at different time points, and then it doesn't add to the accumulation of knowledge. Because if we decide on our own to collect data, I'm not sure that's going to really help in the long run. So any guidance on that would be, I don't know who formulated that question. Maybe it was you, Challa. So what could the breeding industry do to accelerate your activities?
Yeah, I have an idea that maybe the breeding industry could provide samples from animals which are given certain diets, and if they're prepared to modulate those diets, that's not so, those feeds, maybe that would provide materials for us to do further studies on tissues of interest, but maybe also the immune system as well. I mean, the gut would be of interest very much, I think, but also the immune system. But that's a sort of cooperation
but in that case, I would say I would think it would help if you then could be more specific and say what we are looking for is animals that are treated in different ways. And if you look at where we do have animals, they're kept on pretty different diets as well. And then I guess through EFAP, those populations might be available already. The other question is, are we willing to provide different feeds? That's a different question. But already now there might be populations that are given quite different feeds. But then I think you would need to formulate the question a bit more specific into what are you looking for? What kind of diets?
Yeah.
And then there is there's an extensive network, and if it's about sharing data, that's because there's vast amounts of animals being kept by the breeding organizations. And if it's a matter of sharing, that can be organized. If it is giving animals specific treatments, then that's quite a different ball game, I would say. Yeah.
I see. Yeah, and we would also need fecal samples as well, I guess.
Yeah. Okay.
Johan, there are two raised hands by Elizabeth and Anna.
Yes, I wanted can I?
Yeah, go ahead. Go ahead.
Go ahead. Yeah, thank you. So about what can breeding industry do, just out of my mind, I think a thing that is going on since always, it's the IP issue. And I think there is a strong need to really work sensible solutions for the sharing of data and, for example, annotation data for the future. This is something which has been worked on, and it's also in the plans for the Fang data portal and for the development in the future of Eurofang. It's also part of an application that we have submitted for a new infrastructure. And that, I think, will be a key point to really share the same references for functional annotations and then build up on that. And then I was thinking another two things, always in the genotype to phenotype equation. Of course, maybe really understanding for these new phenotypes, for very complex traits that are emerging and important for sustainable productions, to really work together to a better definition of these phenotypes and how they are linked to the intermediate phenotypes that we can measure more easily at a molecular level. And that, of course, is another big thing which is part of much of research right now, but I think in which breeding industry has a leading role. And another thing that I was thinking was about the biorepositories. There are ways to make repositories that are well known by many people and the industry as well, frozen samples, etc., that has been done for decades. Nowadays, it's also possible with a little effort more to store tissues in a way that they can be actually cryopreserved and may be used for the development of in vitro systems for further research. And that is something that if it is done on key animals from specific lines, breeds, important animals, could be a very promising avenue for the future where, again, industry and academics could interact. I'm finished.
Yeah, I hear what you say, and I but if we look at in particular at the poultry sector, we need to be well aware that is basically that's a two-player market. So if you say, well, could you make, as Hendrix Genetics, could you make the DNA available of your top animals of a wide layer? Then, yeah, you can ask a question, but I'm going to tell you, well, sorry, but we're not going to put that into the public domain without any constraint on it, because that's too important for us.
Of course, but
so that's but that's where we need to realize that sensitivity, because and that's not the same as saying that we will never, because we have just also agreed that we will contribute to the initiative to collect poultry genomes. That is fine, but we need to also find that solution where you indeed provide the research community, which is important for all of us to advance. But on the other hand, recognizing that it's not only about generating scientific advancement, but it's also about protecting IP that has been generated by, and then I'm not talking about patents, but just breeding stock that is the generations have spent time on that, generating it.
I can say, of course, I mentioned the IP issue, not to say that it is a stupid issue, the contrary. It's something which exists because it has a reason to exist.
Yes.
The point is that if you think of centralized platforms like Fund Data Portal, among others, there are ways to handle information and pieces of information at a different level of sensitivity, with different access, with different features. It's not that you can simply, that the idea is simply to share everything. We would be quite innocent to think that. So the future is like to work better on this issue to really exploit at maximum what can be exploited by both sides. And maybe one side can exploit more in one sense and the other side can exploit more of the other.
No, so I also did not mention it as, but this is a no-go area, because at the end of the day, all breeding organizations, well, they're clever altogether, but we need the collaboration with research institutes, university in Europe and across the globe to address all the challenges around. So we need to, and we in the past we have found solutions to exchange information, and we will continue to do so. But I said, okay, yes, but it's good to be aware of that. If you say, oh, of some unique animals, then I think we touch a sensitive area.
Yeah.
Okay, Anna, you raised your hand as well.
Yes, thank you. Good afternoon.
Good afternoon.
Well, the point of the data you have already talked about, so you don't need my inputs, you know. I mean, you were already discussing just to, I was thinking there are other breeding companies today around in this webinar. So perhaps it would be good to hear from them. But I was thinking you were mentioning Guelph session. So perhaps FF can, I mean, you were just saying that FF can contribute to contacting breeding companies and see how they can contribute, or what they should expect from this kind of projects. So we can, of course, take contact with breeding companies ahead of the WC Guelph. So starting preparing, not just arriving at the WC Guelph and saying, hey, we would like to have this and this and this. So I don't know in which way, but we can think about it. That's my first 2 cents of today.
No,
the second one.
So,
Yeah.
so Anna, if I can, so what the discussion was, Jerry said, oh, I would like to have this or that type of data, and IFAP is involved in the gene switch project. Then I said, okay, then maybe IFAP can contact breeding organizations with a specific question. Would you have data like this available that you would be willing to share? And play that intermediate role. And yes, if
yes, but
it would be good to do that prior to WC Guelph and not at WC Guelph. Yes, now I fully agree.
Yeah, ahead of the WC Guelph. But I was thinking if it's relevant to, depending on what these breeding companies are answering, to I mean, this WC Guelph is going to be the opportunity to exchange in person. So depending on the question, on the answers given by the breeding companies, if around this gene switch afternoon on Sunday, we can go further. I mean, not me, of course, but the gene switch partners with the breeding companies that have answered this question here. That was the aim. The second one is that we are organizing every year challenge session at EAP, and Filippo is aware about the topic of this year is this data thing you were discussing, Elisabetta and you, Johan. So one of the speakers we were reflecting about within the Fiber TP board was the Eurofang, was from the Eurofang consortium. So perhaps we can address, because from the dairy and the ruminant sector, we have already the speakers, but perhaps we can, that is a follow-up of the previous year conversation. Who can use this data, how we can protect data, but take also the knowledge from having different sources. So yeah, we can, whatever it is, Mario or Elisabetta, or we can think about introducing from the monogastric side this topic on this Salion session in Porto this year. Filippo, I don't know if you have something to add. You know the program of our session.
Excellent comment. Appreciate you advertising that as well.
Yeah, that's indeed very good. If you then mention where it is and when, then I go to the other question of Mario.
It's going to be in Porto, and it's going to be 5th to the 9th of September in Portugal. Yes.
There we go. And the climate is pretty good at that time, probably as good in Rotterdam in the middle of the summer. Mario, you had your hand raised.
Yeah, so just to follow up on the gene expression, like you said something about what to measure or how, or so I think it's good to mention that in this mouse data, actually the gene expression was measured in the liver, but we saw that for all the traits we looked at, it explained quite a bit of the variance. So I think to think about implementation, it would be good to have an idea about what would be reasonably easy tissue to measure on a fairly large scale, preferably in a non-invasive manner, right? So to try and approach it from both sides, like from not only from what is the perspective, what can it do, but also from the application, what would be possible in terms of, and then try to think about where does that match, because seeing that measuring gene expression in the liver explains variance in a lot of traits, perhaps just suggests that wherever you measure gene expression, it could be related to a lot of traits.
Yeah, and I think, Mario, thank you for also reiterating basically that the work done on model animals, like mice in this case, I think it's a compliment to you and the team to analyze the data, because it might be indeed guiding us where to go in terms of measuring gene expression rather than going to each organ of interest, because there's quite a number of them. Okay.
Johan?
Yes. Go ahead.
Just staying on the same topic of breeding company and research, I always see three options, which, the first option, breeding company just, provides some funding to researchers to do the research. The second one is an exclusive research of the breeding company with a certain group, and this provides, NDA, and the research is only, the outcome only is proprietary for the breeding company. And then, which I think goes back to Anna's comments, there is a possibility of creating a pre-competitive space where it's, you can, more than one breeding company can be part of the research, and, you maintain a certain type of confidentiality, obviously, but, you tend to share, you need to do the research together with others in a sense, because it's just, you cannot do it alone in a sense. And I say an example is the feed efficiency on the dairy side, that, a lot of breeding companies kind of join forces to collect the data across different countries. So as long as it's a pre-competitive thing, so can create that space for the breeding company, will you feel more comfortable here on and if there is an option for that in a poultry, which is only two players, as you said. So it's maybe.
Yeah, but I think it's good that there is various ways, and I think and I think at the end of it, I think it is important that we keep the dialogue, what is best, and also keep on the good practice, because I think I do see a lot of collaboration, and I'm really pleased to see that. So it's and yeah, different formats that you mentioned, that each of them has got their advantages, but also their disadvantages. So it's for the given call or subject, make it fit.
So
I as moderator, I also need to keep an eye on time.
Yeah. Johan, I have 2 questions for
Yes.
the audience again.
Yeah.
Can I share them before you go to closing?
Yes, that's good.
So feed companies should invest more effort into dietary studies on microbiota and animal health. One is more clear. I think everybody's clear on this. Okay, so it's a big yes, of course. And the other question is to the breeding companies. Should they consider impact of different diets on selected traits and phenotypes? So, yeah.
Oh, that's a unanimous score. That's
yeah.
Yeah. Well, and I think that's indeed, and if you would have asked me to predict, I would have. I think this is a highly relevant topic to feed the world in a sustainable way and adapt to all the changes we need to do as genetic companies, that in close coordination with what is happening on the feed side. Feeds are changing, so that interaction is indeed, we know for long, but that's key. Okay.
We have one hand raised from Jaap.
Yeah,
Dougie, yeah.
Dougie.
Okay.
Yeah, I'm changed myself back to a human being. I was one of them who responded, I'm not sure if feeding companies should invest more in the microbiota. What is worrying me a bit is we are generating a lot of data with this. And to what extent does it really help us to understand what's happening? Because data collection has become so easy, and you see many responses in terms of microbiota, in terms of, as Jerry told, short chain fatty acids. But what does it really mean? And can we make sense out of this? And my suggestion would be that there should be more money in trying to get this whole picture together rather than investing massively in data, because we have to understand how this very complex system of an animal with its microbiota and its environment operates. And sometimes it worries me that we are putting a lot of effort, because it's easy in data collection, without trying to really understand what's behind it.
I would have I understand the question was not posed to me. I think that
no.
if you look at the date of finding genes responsible, it's really helped to have huge data sets. But huge data sets without a clear hypothesis behind it, we all know that's pretty useless. So what I liked about one of the things I liked about Jerry's presentation, these were the hypotheses. And then you start collecting data.
Yes.
Because if you just collect data for the sake of data collection, yeah, there's always an answer in it, but maybe not to the question that you had in mind. So
if there was a question in mind.
But I think to your point, we should. It's important to collect data, but let's focus at the balanced question and data to answer the question, and not forget that question part. And the question part is maybe more difficult.
Yes.
Than generating good hypothesis is not trivial. Well, collecting data is also not trivial, but that's okay.
Jerry has had his hand raised.
Yeah, could I respond to that, Johan?
Yeah, sure.
Yeah. Can.
Okay. No, I totally I agree, and it's a sort of yes and no for me, because on the one hand, data enables you to look at correlations and associations, but it doesn't provide all the evidence you need to conclude, that a certain group of bacteria is important, and that you might be able to use that information in a valuable way. And that's the same situation in animal health, in human health, that you've seen. There's been a lot of cohort studies, a lot of microbiota collection and data, and it really, there has to be a change, and it is happening, that people now start to have hypotheses and use models and experiments to really try and work out what is happening and why certain bacteria are beneficial and symbiotic, and what are the mechanisms. So I think you need both. And maybe there is a point where you perhaps have enough data and you need to have some hypothesis to look at. But it has been valuable in humans. So, I mean, I really like the work of Ken Honda's lab, because he looks for groups of bacteria that have certain effects on the host. So, he can eliminate a bacterium like Klebsiella, which is a problem in inflammatory bowel disease. He's also found that a certain Clostridia group of bacteria induce regulatory T cells and suppress inflammation, even when you give them very early on to animals. And they're all mouse models, but and there are also a relationship between the microbiota and cancer treatments now. So that would be very good to work out what is going on there. Are they infiltrating? In the case of colon cancer, are they infiltrating and stimulating an immune response to the tumor? I think you get a lot of ideas from collecting data and seeing the associations. So I don't want to knock it completely, but I totally agree with you that it's very much needed to actually generate a hypothesis and think of ways to test them. I mean, that's so important. It's not just about collecting data. But thanks for the remark, because it's a really good one and it's important. Yeah.
Well, thank you, Jerry. With that, I would like to end the discussion session, which I appreciate. I've probably done a poor job in inviting all the others to add, but okay, that's thank you for having questions and willing to discuss. And I hand back to Elzbieta to say the closing words and probably the announcement for the next gene switch activity. Elzbieta, back to you.
Back to me, did I understand?
Yes.
So it was nice. It was quite of a first for us. And yeah, I mean, I really appreciated the discussion. So I thank you very much, the people who gave talk, and Johan and Filippo for the moderation, and of course the FAB for the nice organization of this event. And as we said, we look forward to WC Guelph conference. And we hope that the people who are here today will come to listen to the talks, and hopefully also for the stakeholders, for the breeding companies, for those who are here to come seeing us on Sunday for July PM for the Bitcoin or genomics, what is best, what is more, I don't remember the title now. Anyway, it's Bitcoin or genomics. So thank you to all also for listening.
Yeah, and once again, thank you as GeneSwitch Consortium to organize this. I really would recommend you, and probably that lands on your desk, Ancella, to evaluate, because I think this is a very effective way. Short, not the entire consortium, but to have that interaction with potential users of results. I highly appreciated it, and well, I would recommend you to follow up rather than organizing a big event in Brussels, because it always comes with travel. This is a very effective way, but it's probably good to evaluate the results. But thanks again to all involved in the organization for putting this together, and for all the people attending. Thank you for your attendance, and look forward to see you in Rotterdam and or Porto.
Yes,
Thank you.
Thank you.
Good afternoon. Thank you.
Thank you.
Thank you.
Thank you very much. Bye.
Bye-bye. Thank you.